speaker is Dr. Ben Bahr from the University of North Carolina, Pembroke, and positive lysosomal modulation to treat age-related protein accumulation diseases is this topic of his talk.
Thank you very much. I'd like to thank Aubrey for inviting me to Cambridge as a late addition to this important and unique meeting, and to all the collaborators and organizers that made this happen. It's important to understand how protein accumulation events lead to compromise of neuronal function. We've been studying how transport pathways are disrupted during protein accumulation events, and the eventual synaptic pathogenesis that's thought to be the best correlate of cognitive decline in protein accumulation diseases. There are several clearance pathways that are involved that ensure that mild protein variants are cleared out so they do not persist. David nicely described the autophagy system. There are several groups looking at the proteasome system as a therapeutic strategy. Dr. Bates at King's College in London described how compromise of these systems, whether through brain aging or through specific proteins that can occur in diseases such as Alzheimer's and Parkinson's, leads to stress to these systems and allowing these occasional folded variants to persist, contributing to cellular dysfunction, adding to what's already happening in protein accumulation diseases. Today I'm going to show you the work that we've done in lysosomes. And I should point out, Aubrey suggested some time ago of inserting enzymes into lysosomes and making them more efficient to degrade the protein accumulation events. We had similar ideas at a pharmacological approach, looking at a class of molecules that act as positive lysosomal modulators. The proof of concept molecule I'm going to show you today is this one, Z-Phe-Ala-diazomethylketone. I will call it PADK for this talk. It's actually a mild inhibitor of cathepsin B and L. And in the protease inhibitor field, most would not consider it much of an inhibitor, as it has an IC50 about 10 micromolar. In our hands, we've shown that it increases the expression of several cathepsins and other enzymes. It appears to be enhancing trafficking and increases the pool of intermediates for efficient maturation. I'm going to show you some in vitro work where cathepsins B and D are increased in a brain slice model over time. And later I'll show you transgenic animal work where you can get a dose-dependent increase for the proof of concept. Now, PADK is not the first paradoxical finding that a protease inhibitor causes increase of protease activity. This has actually been shown for a variety of classical inhibitors, E64, lupeptin, and several groups studying PADK that leads to delayed increases in several different hydrolases in the cathepsin family and other types of hydrolases. One classical study by Sutherland and Greenbaum showed that when they inject very high concentrations of lupeptin in rat, 200 mg per kg, you get the characteristic decline. After inhibitor clearance, you get recovery back to normal levels, but then you see this subsequent increase to over two-fold increase in these enzymes. And what these protease inhibitors might be doing may be related to pharmacological chaperone activity that's well known in the glycohydrolase inhibitors. And also there's evidence in the literature for some time now that during disease states, there's evidence of mild lysosomal modulation occurring. In Alzheimer's disease, as Randy Nixon and colleagues have shown, that there may be a compensatory response, a battle between the pathogenic process and potentially a protective mechanism of increasing lysosomal capacity to offset the protein accumulation events occurring. And you can even see it with the A-beta peptide of Alzheimer's as well. And what I'm going to show you is how we can try to test PADK's ability to offset these types of pathogenic changes that can occur with either A-beta in transgenic animals or with chloroquine in the brain slice model, using chloroquine as a lysosomal disruptor, which disrupts the pH gradient. And actually, chloroquine also has evidence of a compensatory response to lysosomes, but much smaller than what's happening with PADK. So the brain slice model is hippocampal slices in culture. They maintain the cellular organization and synaptic circuitry. In this cartoon, we can look at the types of synaptic markers that are reduced in Alzheimer's and other diseases. I'm going to show you the pyramidal field and the associated dendritic field. This is staining with antibodies to paired helical filaments. In control tissue, you don't see any accumulation. After 5 or 6 days with chloroquine, you see characteristic intracellular accumulation that's characteristic of neurofibrillary tangles. It's polar, it's in each cell. It's on the side of the cells where axons initiate. If you look at the associated stratum radianum, you notice the synaptophysin staining for synaptic terminals is greatly reduced in these tissue that has protein accumulation. If you put electrodes in the tissue, you see a corresponding reduction. And we've led to this pathogenic cascade idea that you're going from protein accumulation, initiating microtubule destabilization that leads to transport failure and an eventual synaptic compromise that's thought to explain, the synaptic dysfunction is thought to explain the cognitive decline. So this is the cascade we think that's part of the process in age-related diseases. Lysosomal dysfunction occurs with age and may be the major risk factor for certain disease states. Chloroquine is our model for disrupting lysosomes. We believe tau is involved, being taken away from microtubule, their typical function on microtubules, being drawn into paired helical filaments. We can monitor the change to microtubules by staining for acetylated tubulin. That's a marker of microtubule stability. You see it goes down with time with chloroquine. Correspondingly, you see an associated change in the synaptic marker. Other events also can contribute to transport disturbances. You can put a spot of horseradish peroxidase on a small zone of the pyramidal field and see the dense arborization of HRP being transported along the dendrites and axons. With chloroquine, you see uptake of the HRP into cells, but very little transport. We know this is microtubule based. We can put on microtubule stabilizing agents and prevent these type of changes. But we'd like to act earlier in this pathogenic cascade to look for a way to offset the changes. Can we promote lysosomal function to eventually prevent the type of protein accumulation events that lead to transport disturbances and protect synaptic integrity as part of to treat the cognitive decline? Now, to summarize about 10 years of work in our brain slice model, I'll just show this cartoon of the pyramidal fields. When we put on chloroquine to block lysosomal function, we see those polar deposits in different types of cells, whether the granule cells or pyramidal cells. In every case, they're always on the side of the neuron where axons initiate. This is associated with loss of synaptic integrity. We have less staining, less electrical signaling. We can monitor synaptic decline with various pre- and postsynaptic markers. When you remove the stressor, remove chloroquine, there's no evidence of overt recovery. However, when you apply PADK, the lysosomal modulator, you see dramatic enhancement of the synaptic markers. Associated with that PADK treatment is clearance among the cell fields, and transport mechanisms are turned back on. This is some of the staining of chloroquine. Chloroquine's put on for 6 days, and then followed by 2 days of vehicle, and you see the persistent polar accumulation. During that 2-day process, you can see recovery and clearance. The pre- and postsynaptic markers go down with time and are recovered with the PADK. Chloroquine causes us a small increase in cathepsins. That's the compensatory response I mentioned. But PADK causes a much more dramatic increase. It's 8 to 10-fold in the slice model and even higher in the transgenic animal work I'll show you in a moment. Quantitation of transport and synaptic markers, you see the corresponding protection. In recent work unpublished, the protective effects seems to go beyond neurons. We see effects in oligodendrocytes where myelin markers are reduced with the chloroquine treatment, and PADK protects that as well. And other groups have shown that microglia might be involved in taking up certain protein oligomers and taking the lysosomes, so we're looking at the microglia as well. We're doing a lot of work on mechanisms of lysosomal modulation. It's been studied with lupeptin and E64 for decades, and people are still trying to figure out exactly how this is happening. Increases rate of synthesis. We've seen that at the message level. There's evidence of increased processing and trafficking, where PDK causes a reduction in early endosome markers, the RAB5, and a corresponding increase in late endosome markers. So there's a trafficking influence of helping the maturation process, and may even help trafficking of the proteins themselves that need to get into the lysosomes for clearance. This is associated with seeing very big increases in active cathepsin B. There's even an increase in the mannose-6-phosphate receptor. We're looking at decreases in turnover of the enzymes, and there's also work being done where you're disrupting protein aggregation events, allowing smaller oligomers to be more efficiently brought over to the lysosome, and there may be even events that can occur outside the cell independent of lysosomes that help take up certain types of oligomers. In vivo, you see a small increase in the pro form of cathepsin B after IP injections of PDK, and a dramatic change in the mature forms. And in vivo work is being done in transgenic animals of Alzheimer's disease. We chose 2 different models because we wanted to test this idea at different levels of the pathology. Perhaps modeling early stage Alzheimer's with a 10 to 11 month animal that has the Swedish Indiana mutations of amyloid precursor protein, versus an animal that's a double transgenic with the Swedish mutation as well as a pre-senilin-1 mutation, and much older. These animals receive daily IP injections of PDK for 9 to 11 days. What you see when you look at the hippocampus and pyramidal neurons, the green staining is cathepsin B, and you see increases in staining. You can measure the optical density of the staining, and you see an increase. If you actually count the number of cathepsin B positive organelles, we do not see an increase in the lysosomes, so we have an apparent increase in content of cathepsins in the lysosomes. It's a specific lysosomal enhancement, very selective, since other types of enzymes involved in clearance are not changed. Neprilysin, insulin-degrading enzyme, alpha-secretase, all these things are unchanged. And LAMP1 is not changed, so we do not have a dramatic increase in LAMP1. We can show co-localization of the PADK modulated cathepsin B with LAMP1, so it's being trafficked to the correct location. When we isolate lysosomes from these animals, from different brain areas, we can show that the lysosomal fractions exhibit an increase in the actual activity of cathepsin B, and the non-lysosomal fractions do not show any evidence of an increase. Now, when we look at the early stage type pathology in these 10 to 11 month animals, and stain them for 6E10, which labels A-beta and other species, the wild type shows no staining in the cell fields. The transgenic shows both cellular staining and occasional extracellular deposits. With PDK, you see the cellular staining has been reduced, and there's no evidence of extracellular deposits anymore. We confirm that the staining inside that pyramidal field is indeed intracellular A-beta 1 to 42. We have antibodies that are selective for that version of the peptide, and that intracellular staining is reduced by the PDK treatment. Of all the species labeled by 6E10, whether it's the secreted forms of APP during cleavage, nothing's reduced by PDK except for the A-beta peptide. This brings up the idea that exactly what is PADK doing, because there's some evidence from other groups that cathepsin B can be acting as a beta secretase inhibitor. So it's been asked several times, if PADK is a mild cathepsin B inhibitor, is it really reducing A-beta by acting as a cathepsin B positive modulator, or is it acting by being an inhibitor of the beta secretase? And in the second animal model we've tested, if we were blocking beta secretase activity, we would have seen a change in that beta secreted form of APP. And again, just like in the previous animal model, there's no change there. The only species we see reduced is the A-beta peptide. And we also tested beta secretase activity specifically. At PADK, nor does another cathepsin B inhibitor block the beta secretase activity kit we use, whereas the typical beta secretase inhibitor shows inhibition. This shows the cathepsin B activity, that PDK is indeed a mild inhibitor compared to your typical nanomolar level cathepsin B inhibitor. So in that second animal model, now at higher levels of A-beta pathology, we see the characteristics extracellular deposits that you can see with H and E staining, and they're reduced by the PADK treatment. When you do 6E10 staining, you can confirm that the 6E10 labels both intracellular and extracellular deposition, and both of which are reduced by the PADK treatment. So there may be some type of equilibrium process. When you reduce A-beta material inside the cell, there's less of it to be secreted to form extracellular deposits, and that's been speculated in other groups as well. We can do double labeling with antibodies to the 1 to 42 A-beta peptide and cathepsin B. And in the transgenic pyramidal neuron, you can see intracellular deposition of the green staining of the A-beta peptide and the normal typical level of cathepsin B. In the same type of transgenic animal injected with PADK, you see an increase in the red staining of cathepsin B and reduction in the A-beta. Occasionally, you see co-localization, presumably A-beta being degraded inside lysosomes by the enhanced cathepsin B. We tested what exactly is happening. Is there a truncation event? So we monitored the levels of not only the 1 to 42 peptide, but the shortened, less pathogenic 1 to 38 version of the peptide using selective sandwich ELISAs. And in both animals, both transgenic models, you see a reduction in A-beta 1 to 42, and correspondingly, you see a small increase in the 1 to 38. So it appears to be that 1 to 42 is being degraded by cathepsin B. We show co-localization. We have antibodies to the 1 to 42 and 1 to 38 version, and you see co-localization. And this fits well with what's been shown by Muka's group in San Francisco, where they actually genetically upregulated cathepsin B in transgenic animals and saw that same type of effect of reduction in cathepsin B. And he did the pure experiment of putting pure cathepsin B and pure A-beta 1 to 42 in a test tube and sees production of the 1 to 38 and continued degradation of the peptide. We've done several behavioral experiments, learning after several trials to negotiate a balance beam, remembering the second time being in an open field environment. There's reductions with the transgenic phenotypes and protection by PADK. In the rotarod model over trials, wild types show show improvement across trials. Transgenics show no improvement. With PADK, the transgenics are now able to improve over trials and actually are now statistically better than the wild types without PADK. And remember, these are aged animals that may be experiencing mild age-related synaptic decline, so PADK might be influencing activity even over the aged wild types. When PADK is added to wild types, there's no effect, and across the different groups of animals, there's no change in the basal mobility. And there's a correspondence between behavioral protection and synaptic protection, showing that link between synaptic pathology and cognitive decline. In the spontaneous alternation behavior, where animals are searching 3 positions for food, and alternation is where they don't go back in the same place they just came from, there's a reduction in the transgenic animal, improvement by the PADK. Correspondingly, there's mild reductions in synaptic markers in both animal models, and PADK shows recovery, particularly in the postsynaptic glutamate receptor from AMPA receptors. And if you look at the staining of that subunit, the reduction appears to be in the dendritic fields, and corresponding with changes in synaptic integrity that you can visualize with calbindin staining that also shows evidence of protection. Now, as a preview of a poster that's out there for my postdoc, we're now developing non-peptidyl derivatives of PADK to make more drug-like molecules because the peptide bond in PADK has stability issues. PADK is a mild inhibitor, as I mentioned. The concentration we usually use for modulation is 1 to 5 micromole in the brain slice model. So you're working at a level that might be causing mild inhibition. But now we've made derivatives that are decoupling this inhibitory activity of these molecules to focus more on the modulatory activity. And we made several families of these compounds that are more potent than PADK by monitoring cathepsin increase in the slice model. One particular derivative, BW1002, is more potent than PADK and also been shown to reduce chloroquine-mediated protein accumulation in the slice model. Now, before I close, I'd like to say any group that's working on these types of enhancement of clearance mechanisms need to consider the pathogenic stage of which you're putting on the drug. David alluded to this, and we know that the aging brain is up against a variety of changes, whether it's aberrant proteolysis, reactive oxygen species, all of which can lead to lysosomal stress. And cathepsins in particular, when you talk about the kind of drugs we're looking at that enhances cathepsins, they've been implicated in being released from lysosomes and actually contributing to pathogenic processes, whether it's mitochondrial dysfunction, apoptosis, and further oxidative stress. Early on in early stages of disease, even in early stage Alzheimer's, it's been shown there's activation of the autophagy lysosome system. And this may actually be trying to hold off the pathology, reducing gradual protein accumulation that leads to the synaptopathogenesis. Perhaps that explains why Alzheimer's can last from 2 years to 15 years in certain patients of this battle between protection and pathology. So in closing, to sum our current ideas and our future ideas, that lysosomal modulators can enhance enzymatic clearance, to have intracellular clearance, perhaps through cathepsin processing of different proteins, whether it's tau or A-beta. There could be disaggregation occurring to allow smaller oligomers more efficiently traffic to the lysosomal pathway. There's also ideas of using drugs that act extracellularly to disrupt aggregation to allow smaller oligomers to be taken up into neurons. And we've shown neurons, particularly CA1 pyramidal neurons, take up A-beta and sequester the peptide into lysosomes. And more recently, other groups have shown microglia are involved in taking up A-beta. We also have new data we just published of an idea of, independent of lysosomes, pro-aggregation of pathogenic oligomers, where you have oligomers on the outside of the cell that are pathogenic, the soluble oligomers that have been thought to be the biggest contributor to the disease, where you can actually promote their aggregation into larger complexes that are now less pathogenic, sequestering them into a benign state. And that's actually a debate whether plaques are doing that themselves. All of which leads to stabilizing synaptic integrity, leading to normal information encoding processing, and actually repair signaling that's occurring through synaptic communication. So I'd like to thank the members of my group. Dave Butler, some of this work was his PhD thesis, and then he stayed on as a postdoc and did more work before he got taken away by Harvard. My current postdocs, Megan Wisniewski and Vino Naidu, have taken up the reins. Vino, sadly, well, happy for him, sadly for me, he just moved back to South Africa, I think yesterday, so he won't even be there when I get home. Collaborators doing the PADK derivatives are Dennis Wright at University of Connecticut and Dennis Hoover of Synaptic Dynamics. Thank you for your attention.
This paper is now open for questions.
Very exciting stuff. You said you had unpublished work on testing its effects on oligodendrocytes, and also that you were starting to do work on the effects on microglia. Would you care to elaborate briefly on what you saw in the oligodendrocytes and what you're looking to do with the microglia?
The oligodendrocyte work you basically saw, it's very early work of treating hippocampal slices with chloroquine, and that causes a dramatic decrease in that myelin proteolipid protein. And when we applied PADK for 2 or 3 days, it looked like normal levels were returned with the PADK treatment. The microglia, we're currently buying the appropriate antibodies to look at co-localization of microglia markers and cathepsin B in the hippocampal slices to see if we see indications of increased cathepsins. That's where we're going to start. So we actually have no data yet on the microglia.
And none of this is with A-beta yet. This is all just with chloroquine.
This is all with the chloroquine model. We always start with that. We can actually look at some of our transgenic animals just with the microglia staining and try to pull out some tissue out of the freezer and do some staining. So that's in the works as well.
Thank you. We are out of time, so I'll move on to the next speaker.